Hello, biologists. Today we're going to talk about cellular respiration in detail. Let's start with an overview so we can see where we are, where we're going, and what we're going to discuss. The first step of cellular respiration is glycolysis. This is step one. That occurs in the cytoplasm. In glycolysis, you are taking glucose that you consumed, you ate it, whatever it is. Most things have glucose in them in some way. And you are going to break glucose into two pyruvates. Each of those pyruvates is going to go through the next step, but we kind of look at them one at a time. So just remember that there's two pyruvates. So you do two processes through this whole thing per glucose, not just one. When you do glycolysis, you're also going to produce 2 ATP, and you're going to produce 2 NADH. NADH are those high energy electron carriers. The electrons have a ton of energy, and we're going to use it later. The carriers pick them up and transport them to where they're going to go. From glycolysis, they have to get from the cytoplasm into the inner membrane of the mitochondria, so they're going to get moved inward. So we can follow those to NADH. The next step of cellular respiration is for the pyruvate to get into the mitochondria, and we're going to do pyruvate oxidation. In pyruvate oxidation, we're going to take pyruvate and we're going to turn it into acetyl-CoA. Remember the acronym oil rig? Oxidation is loss. Loss of what? Electrons. So pyruvate oxidation means we're removing electrons from pyruvate. Where are those electrons going? They're going into their high energy electron carriers. And then the second part of the acronym is RIG. Reduction is gain. So oxidation is loss, reduction is gain of electrons. This is often referred to as redox reactions, oxidation reduction. So just be aware that people say that as well. Pyruvate oxidation, we're going to remove some electrons, put them into their carriers. In the process, we're also going to produce carbon dioxide, which is a waste product. Acetyl-CoA can now enter, this is step 2, by the way. Acetyl-CoA can now enter the Krebs cycle or the citric acid cycle. Those are two synonyms. You can use either one. This is step 3 of the process. In the citric acid cycle, we're going to start with an enzyme. It is called oxaloacetate, a great enzyme. I like to abbreviate it OAA, oxaloacetate. And that is the beginning and ending molecule in the citric acid cycle. Oxaloacetate binds to acetyl-CoA and goes through several transitions. And as it goes through these transitions, it produces carbon dioxide as a waste product. It produces ATP, but the real point, like the big deal of the citric acid cycle, is it produces several high energy electrons, and those are picked up by the carriers, and we're taking them to the last step of the process. At the end of the citric acid cycle, you're done with all the carbon dioxide that used to be in your glucose, and you are going to regenerate oxaloacetate so the process can begin again. That's why it's a cycle. Now, all of these electrons that are so full of energy are going to get taken to the last step, which is the electron transport chain. That's going to be step 4. The electron transport chain is where we're going to make the bulk of our ATP. So a huge amount of ATP, which is our overall goal of cellular respiration. We need to make that cellular energy. And so this is the heavy hitter in the entire process. How are we going to make that ATP? We're going to use oxidative phosphorylation. That is the process of using an enzyme called ATP synthase. To synthesize something is to make it. We are making ATP. ATP synthase makes ATP. The electron transport chain is going to set everything up that we need to use ATP synthase. So the electron transport chain is going to be used to make a concentration gradient of hydrogen ions in between the inner membrane and the outer membrane of our mitochondria. And the process of those hydrogen ions moving through ATP synthase enzyme is called chemiosmosis. It's like osmosis, but with chemicals. So think chemiosmosis. It's the movement of those hydrogen ions. Those hydrogen ions are going to power ATP synthase, which is going to make us all our ATP. And none of this could happen without our oxygen that is necessary for aerobic respiration, because the oxygen allows the the enzymes in the electron transport chain to function. When we use up the oxygen, we're going to reduce it and produce water that's a waste product. So that's our big overview. Give me a second to erase all that, and then we will go through each step in detail. As we go through in detail, I want you to think about, what does each step start with? What does it end with? And what's the goal? Like, what are we trying to do with this step? Our overall goal is to produce ATP, but we won't see that in every single step. Here's our beautiful mitochondria. The shape of the mitochondria is important to its function. So mitochondria have two membranes, the outer membrane and the inner membrane. It got that second membrane during endosymbiosis, which you read about during week 1, where the original prokaryote bacteria was engulfed by one of the first cells that became a eukaryote. And mitochondria themselves used to be free living. They're about the same shape and size as bacteria. Each cell has hundreds and thousands of them. They also have their own DNA, not your DNA, and they reproduce on their own schedule, not with your cell. So all those things indicate that they used to have a different origin. That's the endosymbiotic theory. So that second membrane actually became really important for us, because we're going to use the space in between the inner and the outer membrane to create a large concentration gradient. And that concentration gradient is going to power ATP synthase. So the function and structure of the mitochondria are completely interlinked. So outer membrane, these folds of the inner membrane are really important. They're called cristae, which reminds me of like a name. Hey, cristae. So cristae. And these cristae help increase the surface area of the inner membrane, and they're completely embedded with electron transport chain molecules and ATP synthase. So the entirety of this inner membrane is working all the time to do ATP production. Inside, you also have little granules and bits of DNA and tiny little proteins, but it's that inner and outer membrane that we really care about for cellular respiration. This is a picture of a mitochondria from real life. This is a transmission electron microscope, which means this is real. And you can see the space between the inner and the outer membrane is quite small, but there is a little gap between them. And all the beautiful folds of the cristae are super long. When we draw them, they look kind of smaller, but they are very long and very numerous. So there's a lot of surface area. And that gap in between the cristae right here, that is intermembrane space. The intermembrane space continues into the inside of the membrane. So all along here, you have an increased hydrogen ion concentration in those little gaps. So structure and function, super important. Step 1 of cellular respiration is glycolysis. When we do glycolysis, we are taking a 6-carbon glucose. Each of these balls is a carbon, and we are splitting it into 2-carbon pyruvate. To do that, you have to invest energy. So there's the energy investment phase and the energy generation phase. Put 2 ATP into it. You're using the energy from the ATP, which leaves you with ADP, adenosine diphosphate, because you've popped that last phosphate off. That gives you 2 molecules that are intermediary. You need to add phosphates to them so they're stable. And as you do, you can harvest electrons from them, and then you remove those phosphates, and you get a little ATP back, and you're left with 2 3-carbon pyruvates. Overall point, we are generating ATP 2 net because we invested 2 and we got 4 out. And we are also harvesting electrons. We haven't lost any carbon yet, but we've taken 1 molecule and we've created 2 new molecules. I do not expect you to memorize these intermediary names, just as a heads up, but I do think it's cool, which is why I like to show you. The next step is pyruvate oxidation. Here's my pyruvate. It has 3 carbons. Those are those 3 black balls that we just saw in the last image. Pyruvate is going to get transported into the mitochondria using a transport protein. And as it gets transported in, we are going to change it. The way that we change it is we break off one of these carbons. We exhale that carbon as carbon dioxide. That's a waste product. We're going to harvest some electrons. We're going to put those electrons in the carrier. Remember, this is an empty electron carrier, and this is a full electron carrier. Those electrons will be taken to the electron transport chain later. And we are going to add coenzyme A. Coenzyme A being added to these 2 carbons here creates acetyl-CoA. Acetyl-CoA is the molecule that we are going to put into the next step, the citric acid cycle, also called the Krebs cycle. So here we have pyruvate. We have added this coenzyme A. We have removed a carbon, and we have removed electrons. We are left with acetyl-CoA, and now we get to start the the citric acid cycle. Here's oxaloacetate. Oxaloacetate is a 4-carbon molecule. It's going to bind to the 2 carbons left from glucose in acetyl-CoA. As it does, it breaks off coenzyme A, and coenzyme A can be reused in pyruvate oxidation. So it just goes back over to pyruvate to help out in the process. That 6-carbon intermediary molecule is called citric acid, which is why this was originally called the citric acid cycle. Citric acid is going to be broken 1 carbon off. Some electrons are going to be harvested off. You're going to be left with a 5-carbon intermediary. We're going to remove another carbon, exhale it as carbon dioxide. We're going to harvest more electrons and a little ATP, and we are left with a 4-carbon intermediary. Things to note so far: the 2 carbons that were left from glucose have now been removed in our waste products. That 4-carbon intermediary is going to get altered. We're going to take some electrons from it in the form of NADH and FADH2. Those are going to go to the electron transport chain later. And that 4-carbon intermediary is going to be regenerated into oxaloacetate, so the entire process can begin again. Remember, for each 1 glucose, you get 2 pyruvate, 2 acetyl-CoA, and that means that you do the citric acid cycle twice. So we do this twice for each glucose. What are we left with? It looks like everything's gone. We're left with these high energy electrons in their carriers, and we're going to use those in the last step, which is the electron transport chain. Before we get there, I want to show you the beautiful process of citric acid cycle in a little more detail. I don't expect you to know the names of the intermediaries, but they are super cool, and so let's walk through it. Here's pyruvate. These are the 3 carbons that are left over from glucose. Remember, glucose is a 6-carbon molecule. This is acetyl-CoA. We have our 2 carbons and our coenzyme A. To get from pyruvate to acetyl-CoA, we have removed one of those carbons, and it is exhaled as carbon dioxide. The enzyme that does that is called pyruvate dehydrogenase. It's an ACE, so you know it's an enzyme. Acetyl-CoA will enter the citric acid cycle. It will bind with the beginning molecule, which is oxaloacetate. Oxaloacetate binds to acetyl-CoA. Coenzyme A is removed so it can go back and help regenerate the process of pyruvate oxidation again. Acetyl-CoA and oxaloacetate binding together forms citrate, which is created by citrate synthase. Synthase means that it's building something. What is it building? It's building citrate. Citrate is then turned into cis-aconitate. The enzyme that did that is aconitase, which is then turned into 2 more, 3 more, 4 more intermediaries. Each of those is catalyzed by an enzyme that tells you what it does. Look, in dehydrogenase, we're removing a hydrogen ion and a electron again. And then we get down to succinate. And that one is also catalyzed by an enzyme named for it. And then we move on to fumarate and maltate until we regenerate oxaloacetate. But each of those enzymes related gives you the name of what it's doing. And I think that's pretty rad. For us, what do we need to know? We need to know what enters the cycle, what starts the cycle. We do not need to know the intermediary names, but we do need to know what's produced, which is NADH and ATP right here. That's the ATP. It says GTP, but it's the same thing. It's ATP. They're technically different molecules, but they work the same. And so we'll just pretend that that says ATP for us. And in the process, we are done using up all of those carbon dioxides. Those carbon dioxides have been exhaled as waste products. All right, let's get to the electron transport chain. Everything so far has been to get electrons ready for this. Here is the electron transport chain. Let's orient ourselves. This is the mitochondrial intermembrane space. So this is going to be the inner membrane. And then you have the matrix, which is the inside of the mitochondria. So this is a cristae. Good? Awesome. Each of these inner membranes is made of a phospholipid bilayer. Here we have little blue dots. Those are hydrogen ions. And then inside the inner membrane, we're showing you the electron transport chain, which is made of these different proteins. The first protein is NADH reductase. It's reducing. Reduction is gain. So that's oil rig. That means something's going to be gaining an electron. Where is it gaining the electron from? From this high energy electron carrier. It's going to donate electrons. Then we have ubiquinone. We have cytochrome reductase. We have cytochrome. And we have cytochrome oxidase. Those are the electron transport chain, those 5 enzymes embedded in the inner membrane of the mitochondria. And last, we have another enzyme, ATP synthase. What is that going to do? Synthesize ATP. So all of those high energy electron carriers that you have been creating throughout the process of cellular respiration come to the inner membrane of the mitochondria and they donate their electrons. The electron moves from a high energy state to a low energy state. And as it uses up that energy, it is shuttled back and forth between these proteins. And then it decreases in energy and has to be received by the final electron carrier, which is going to be oxygen. So oxygen is standing here waiting to grab that electron as it falls down its energy gradient. And as it does, we combine hydrogen to the oxygen and we produce water as a waste product. The big key here is oxygen is required for the electron to exit the electron transport chain. If there is no oxygen, electrons cannot exit, and that means electrons cannot enter. So the electrons, as they are shuttled back and forth, their energy is used to pump hydrogen ions from the matrix into the inner membrane space. So this arrow is denoting hydrogen ions going into the intermembrane space. This creates a concentration gradient of lots and lots of hydrogens here and fewer hydrogens here. Hydrogen, just like all ions, does not want to be crowded. It wants to diffuse back into the matrix. To get back into the matrix, there is only one place it can go through, and that is ATP synthase. So as the hydrogen moves back into ATP synthase, and again, it's following its concentration gradient, so this is not requiring energy. This part, pumping them into the intermembrane space, going against the concentration gradient, does require energy, and the energy comes from those high energy electrons. As the hydrogens leave through ATP synthase turns, and it grabs a phosphate, and it pops it onto ADP, and it produces ATP. So all of these hydrogen ions move through this enzyme, and every time they move through, it creates ATP. It will create a bulk 34 to 36, sometimes 32 ATP per glucose molecule, which is where we get the bulk of our ATP for our cells. Without that oxygen, you wouldn't be able to put the high energy electrons in, and you wouldn't get this concentration gradient, which means that you wouldn't have ATP. If this concentration gradient were to be destroyed, and if you had an equal amount on both sides of the membrane, there would be no net movement of hydrogen, and you would not be creating ATP. Here's another visualization of the process. I have my proteins embedded in the inner membrane. This is the electron transport chain. I have NADH and FADH2. They are donating their high energy electrons, which are going to get shuttled through the electron transport chain. They donate them at different locations, but they still donate them to the electron transport chain. So I'm not going to ask you to determine which one donates where. Both of them donate their electrons, which move through the electron transport chain. As they move through, they pump hydrogen ions into the inner membrane space, and they are received by oxygen at the end of the electron transport chain, which is reduced into water. Water is the waste product. Oxygen is required for the electrons to flow through the electron transport chain. That's why you breathe. Like, that's what suffocation is no longer having the oxygen for your electron transport chain. So without the oxygen, you die in a couple of minutes, which is wild because what you're really stopping is this concentration gradient. And that's how important this process is to you. You can't live a few minutes without it. So now that we've created this hydrogen gradient inside the inner membrane space, these crowded protons want to go through back into the matrix. They're following their concentration gradient. And as they flow through ATP synthase, ADP is reduced into ATP, and we have produced energy that our cell can use. Here, it's also showing the Krebs cycle. That's great because it's nice to remember that all of this occurring in a very short amount of space, all constantly, all continuously, all concurrently. So it's all happening at once everywhere inside of that compact mitochondria with all those inner membrane folds that you saw. Now, these have all been schematics. This is our best understanding of what it actually looks like. And I know it's a little blurry, but I don't expect you to know that this is going to be called fumarase reductase or anything like that. But I do want you to see the complexity of it, which is why I thought this image was worth showing. Each one of these is a component of a protein in the electron transport chain. We do know their names, we do know their shapes, we do know their functions. We think we found the genes, the code for these proteins, because these proteins all had to be created by ribosomes, which means that there's a piece of mRNA coded for it in the DNA. And so the entire complex is really beautiful. And I wanted to make sure that you saw the beautiful complexity of it. I have a video up on Blackboard for you to check out. I actually have two. The first one is a really nice overview. It's going to show glucose being broken and the entire process. And then the next one is a little more complex. And I know that it might feel a little overwhelming, but what I want you to take away from it is the big picture. So you do not need to know what's called cytochrome C, but I want you to watch the entire process. And my favorite thing is the end when ATP synthase is shown to be moving. So watch it, marvel at it, be amazed by this process. And if you have any questions, email me. As you're working on this and as you're studying it, I put up a practice worksheet for you on Blackboard. Please try and fill it in as best you can. And note that there are things missing. Like right here, this is going to be the first step. It's occurring in the cytoplasm. That's cool. This is the next step. It's occurring in the mitochondria. It doesn't produce ATP, but in this step, we do produce carbon dioxide, and that's not being shown on this. I also think this diagram is missing oxygen. So where is oxygen used? Make sure you add that to your diagram as well. And then when you are done practicing with the diagrams, you can come to this next slide that I'm going to show you, and that has the answers on it for you. But I don't want you to do it until you've tried. The trying is really important. So try first, and then look at the slides that are posted to PowerPoint, or pause the video on this where you can see the overall schematic of it. I hope that was helpful. Email me anytime with questions, and have a great rest of your day.